Welcome back, once again. Today we'll be looking at another course in pharmacy, pharmaceutical microbiology and biotechnology. But for the purpose of this lecture, we'll be looking at the introduction, so bring out your writing materials, let's get started. Microbiology is a broad term, meaning the scientific study of living organisms that are individually too small or so minute to be seen with the naked eyes, without the use of a microscope. Such minute forms of life are generally termed microorganisms, which are sometimes colloquially called microbes, or in the vernacular as germs. Make sure to subscribe while we continue the class. Microorganisms include bacteria, fungi, viruses, protozoa and algae. Hence, microbiology includes the study of this bacteria, fungi, viruses, protozoa with some algae as well as some other forms of life that do not fit well into any of these broad microbial physiological groups. Pharmaceutical microbiology may be defined as the special aspect, part of microbiology which has special bearing on pharmacy in all its aspects. Pharmaceutical microbiology is highly relevant as one of the core courses in pharmaceutical sciences, given the following significances. 1. Microorganisms are everywhere, and hence, are the major etiologic agents of many mild, severe and debilitating infections and diseases of humans, animals and plants. 2. Microorganisms, due to their ubiquity, frequently contaminate pharmaceutical products, leading to microbial growth, causing biodeterioration and spoilage of pharmaceutical products, resulting in the loss of product quality. Hence, a potential public health hazard and risk of infections, diseases on consumption to customers, and products rejection and high economic losses to the manufacturer. 3. Microorganisms, when inadvertently introduced into the body during drugs injections, even in the absence of infections, cause pyrogenic reactions or pyrogenicity, fever, a very serious public health hazard. 4. Microorganisms, particularly bacteria, through various biochemical and genetic basis, frequently mutates in the presence of antibiotics, thereby providing a large continuing reservoir of antibiotic-resistant genes, resulting in the ever-increasing case of the non-mitigated troublesome issue of antibiotic resistance. 5. Microorganisms, given their broad diversity and ubiquity, are the original, major sources used in the manufacture, production of many valuable pharmaceutical products such as antibiotics, steroids, therapeutic, industrial enzymes, fine chemicals, examples alcohols, vinegar etc., vaccines, polysaccharides, products of recombinant DNA technology. 6. Microorganisms are veritable tools, agents usually employed in pharmaceutical productions, as microbial assay organisms to determine antibiotics, vitamins, amino acids, polysaccharides, vaccines. 7. Microorganisms are useful tools, agents, used for the precise and concise defections of mutagenic and carcinogenic activities, as well as toxicity of various pharmaceuticals, prior to usage in therapy. Mutagenic activity is the ability of a substance or agent, called a mutagen, to cause permanent changes, mutations, in DNA. While carcinogenic activity is the ability of a substance or agent, a carcinogen, to cause cancer. 8. Pharmaceutical microbiology knowledge and understanding is highly useful in pharmaceutical products formulation, quality assessment, control and assurance, as well as validation for both sterile and non-sterile pharmaceutical products. Pharmaceutical microbiology knowledge helps in the adequate microbial control in the environment by the use of physical agents, dry heat, sterilization, irradiation, and chemical, antimicrobial agents, antibiotics, disinfectants, antiseptics, preservatives, sanitizers, immunotherapeutics, etc., to adequately, effectively, efficiently control the spread of microbes in the environment, animate, inanimate objects and surfaces, as well as on, or in pharmaceutical products. These are the physical agents while these are the chemical agents. Let's explain what immunotherapeutics is. They are chemical agents used to control microbes indirectly by strengthening or modifying the host's immune response, rather than killing microbes directly like disinfectants or antibiotics. So pharmaceutical microbiology knowledge helps you control the spread of microbes in the environment, animate, inanimate objects and surfaces, as well as on, or in pharmaceutical products. And that wraps up our introduction to pharmaceutical microbiology. This subject is a foundation for understanding drug safety, quality control, and infection prevention. Stay tuned for upcoming lessons where we'll break everything down step by step. Like, share, and subscribe for more pharmacy and health-related content. See you in the next video.